Hello everyone and welcome to the channel of Pharmacology, where today we will be discussing about reverse pharmacology. So without taking much time, let us begin. Now we will see what do we mean by reverse pharmacology. So for your easy understanding, I have just broken down the definition so that you get a concept of the reverse pharmacology from this definition only. So now the first part of the definition, it tells us what exactly is reverse pharmacology. Now reverse pharmacology, it integrates documented clinical or experimental hits into lead compounds. That is, when we are developing a drug, first we have the hit compounds and then we have the lead compounds, and subsequently from the lead compounds the drugs are developed. This is a conventional pathway, right? Now the second part of the definition, it tells us that how is reverse pharmacology going to integrate the hits into leads, right? How they're going to convert the hits into leads. It is going to do so by a transdisciplinary exploratory studies. Now transdisciplinary means that not a single discipline is not enough. So we are going to integrate ourselves to various disciplines of pharmacology, right? And finally, what is the aim of reverse pharmacology? So what is the aim of these two processes, right? Rather these two steps? The final aim of reverse pharmacology is to develop these into drug candidates with the help of experimental and clinical research, right? So this is the definition. So by definition, reverse pharmacology is a science which is going to integrate the documented clinical or experimental hits into leads by transdisciplinary exploratory studies, and ultimately developing these into drug candidates by the experimental and the clinical research, right? So let us move on. Now let us study a little bit about the history of reverse pharmacology with respect to the Indian perspective. Okay. Now in India, the reverse pharmacology, the pioneer of the concept of reverse pharmacology was Sir Ramnath Chopra. Sir Ramnath Chopra is also the father of Indian pharmacology. Sir Ramnath Chopra along with Gyananath Sen, together they laid down the foundation of reverse pharmacology. And Gyananath Sen with Karthik Bose, they demonstrated first the anti-hypertensive and the tranquilizing effect of Rolfia serpentina, right? And this happened in 1931. Okay. Now the question that arises is, why do we need reverse pharmacology? Now you know that there is already a classical approach by which a new chemical entity is ultimately converted into a drug, right? Now this approach is the classical forward approach. But when we go through this approach, first we have to establish the activity of the NCE or the new chemical entity on cellular and preclinical level. After we have established the activity, the safety at the preclinical level, then we are going to move on to the clinical level. And after we have developed, or rather after we have established the same thing at the clinical level, then the drug is going to come into the market. This is the classical approach. But the problem with this approach is that it takes a lot of time. That is the first problem. That this approach, it takes a lot of time. And the second problem is that there is a huge amount of money that is involved. Now with the increasing research cost, that is now the cost of research has gone up very much, and also a lot of new compounds can be screened because of the advent of high throughput screening and all those processes, right? But even after all these things, what we are seeing is that the number of approved new chemical entities, they are actually decreasing. So the cost is increasing, the technology is increasing, but the number of approved molecules or new chemical entities, they are decreasing. So now the pharmaceutical industry, instead of going for the blockbuster drugs, they are now going for a much safer option, and that option is known as the Me Too drugs. So this conventional method is not going to cut it, because the new chemical entities are decreasing, but the diseases that we are encountering now, they are becoming more and more evolved. So we need a process so that we can quickly develop the chemical entities into potential drug. And that is where the concept of reverse pharmacology comes into being. The concept of reverse pharmacology, it takes care of three most important bottlenecks of the classical or the conventional drug approach. And those three bottlenecks are time, money, and toxicity. So these three bottlenecks are taken care by the reverse pharmacology approach. And since the conventional approach is not working, rather it is not sufficient enough, now we need the alternative methods. And one of the most important, or rather one of the most popular alternative method that is now coming into being is the reverse pharmacology method. Right? So what is the concept of reverse pharmacology? Now if we look into the concept of the classical pharmacology, for the purpose of development of a drug, we are moving in the forward direction. Okay, that is, we are moving from the laboratory towards the clinics, right? That is, first there is a target identification, then there is a lead compound, then there is lead optimization, then after lead optimization there will be preclinical studies, then after that, after all the favorable data, we are going to move into the clinical trials, and finally the drug will be there in the market. But in case of reverse pharmacology, we are just reversing this direction. Okay, we are not moving from the laboratory to the clinics, rather we are moving from the clinics to the laboratory. That is, first we are starting with the clinical trial, and then we are coming back, and then we are seeing that we are rather isolating the compound, which can be a potential drug. That is how we are moving in case that is how we are moving in case of reverse pharmacology. So the direction of drug has been reversed, and that is why it is known as reverse pharmacology. The reverse pharmacology, the main aim of reverse pharmacology is to use the drugs that we have been using for centuries in traditional medicines, but not the folklore drugs, the proper scientific drugs that we are using. And the second thing is that it integrates the traditional system of medicine with a safety and efficacy record. That is, there is robust documentation. This robust scientific documentation is very important, because if we use the compound that we have been using for centuries and we have got proper data about its safety and its efficacy, then the number of NCs that a number of new chemical entities from which we can develop new drugs, there will be far more. So that is the main aim of reverse pharmacology. Okay. Now moving on to the scope of reverse pharmacology. I have already told you that reverse pharmacology is integrating traditional system of medicine with proper safety and efficacy data. That means the first scope is that there is clinical evaluation of safety and efficacy of whom? Safety, efficacy and quality. Quality of whom? Of all the drugs or the plants that we are using in the traditional system of medicine. So now in case of reverse pharmacology, what we have is there is a proper and a robust documentation of the drugs that we have been using in the traditional system. So we have a proper scientific data, right? The second scope is that we can discover new drugs from natural products. And these drugs that we are developing from the natural products, these are not folklore drugs, but these are proper and scientific drugs that we are developing, because we have got the data to back up all our claims. Okay. The third point is that through the process of reverse pharmacology, we can overcome the current costly and the lengthy process of drug discovery. As I've already told you that the number of new chemical entities is decreasing, but the cost of research is increasing. So if we already use the resources that we have with us for centuries and properly document everything, then the synthesis of new chemical entities, it is going to take us a lot lesser time. So we are going to save time as well as money. Okay. Now students, what are the phases of reverse pharmacology? So reverse pharmacology classically has got 3 phases. But please remember students that these 3 phases, they are not sequential. But rather these 3 phases, they are mutually exclusive. Okay. Now the first phase is the experiential phase. In experiential phase, the most important job is the documentation. Now documentation of what? Documentation of the biodynamic effects. And whose biodynamic effects are we documenting? We are documenting the biodynamic effects of the drugs which are, or rather which were used in the traditional system of medicines. So biodynamic effects of the drugs used in TS, traditional system of medicines. So now we have got a meticulous record of all the drugs that we were using in our traditional systems. The second phase is the exploratory phase. Okay. So what we are doing at the exploratory phase? We are looking for the target activity. Now target activity of what? Target activity of the drugs that we have taken from the traditional systems. Okay. So we are going to look for what? The tolerability of the drug. We are going to look into the drug interactions. We are going to look into the dose ranging. Okay. So we are going to look into all this. We are going to look into all these matters, and these will be seen both at the clinical, that is on the patients, as well as on the preclinical levels. That is okay, by preparing suitable models both in vitro as well as in vivo. Okay. The third phase is the experimental phase. In experimental phase, we are going to identify and validate the safety of the drug. For identification and validation of the safety of the drug, this is going to happen at multiple levels, both at the preclinical as well as the clinical levels. Okay? And students, please remember that in case of reverse pharmacology, the safety of the drug, this is most important. The efficacy is for validation only, but the safety is most important component. Okay? Now, if we compare classical pharmacology with reverse pharmacology, in classical pharmacology, we are starting with target selection. From there, we are going on to the lead compound identification, then the molecular target identification, then we are optimizing the lead compounds. Finally, there is the preclinical and the clinical trials. But this entire process, it takes, for a drug ultimately come to the market, it takes around 10 to 15 years. But a reverse pharmacology, we have already told you, we are moving from the clinics towards the laboratory, right? So we are starting with the clinical trials, phase 1 and phase 2. Since we already have got the data at the documentation phase only, so we already have the data, the entire biodynamic effect. We are starting from the clinical trials only. From there we can move on to the mechanistic paraclinical trials, then the safety studies, then the molecular pharmacological studies, and finally the large scale clinical trials. So we are starting from the clinical trials, going on to the laboratories. But the most important aspect is that for a drug to come into the market, it takes 4 to 5 years in comparison to 10 to 15 years of the classical pathway. So if you look into here also, we are going from the first one, this one is the conventional pathway, forward pathway. From the target identification, lead optimization, preclinical and clinical studies, we are taking 10 to 15 years. But if we are reversing the process and we are starting with the clinical trials and then moving on to the paraclinical safety study, looking to the relevant signs, and then conducting the large scale clinical trials, for a drug to come to the market, it is going to take 4 to 5 years. And since the time is short, so you have already saved on time and the time taken is short, now you have also saved on the money. Okay. And the third point is that there is robust documentation and there is safety studies. So the toxicity component is also taken care of. So the main three bottlenecks, as I've already discussed earlier, the main three bottlenecks, time, money and toxicity, they can be very well reduced with the concept of reverse pharmacology. Now some of the lead molecules which have been discovered by using the process of reverse pharmacology. The first compound is Rolfia alkaloids. So from the Rolfia alkaloids, as when we are discussing about the history, we saw that it showed antihypertensive effects. So we got the antihypertensive effects. Not only antihypertensive effects, from Rolfia alkaloids we also got drugs such as bromocriptine, levodopa, which were spin-offs from this original Rolfia alkaloid compound only. Soralens, another lead molecule which is used in case of vitiligo. Now we have got the Soralens and we use it in case of vitiligo along with ultraviolet radiation therapy, and they are working very well. Next we have got the gogolsterones, the gogolipids, and we use it as hypolipidemic agents. we have got piperidines, which are used as bioavailability enhancers. New drugs such as curcumins, which we have found that they have got anti-inflammatory potential. And now curcumin are also available in the market. And the most and the best example of reverse pharmacology is perhaps Artemisia annua, from which we have got the artemisinin compounds, which are currently the mainstay of anti-malarial drug. And recently, Nicanthes arbotristis Linn, this plant, it has also shown anti-malarial activity. Now a few examples of the drugs which have been made indigenously in India with the help of the reverse pharmacology. As I have already told you, gugulipid. This gugulipid, it was developed jointly by ICMR and CSIR, and it was developed from Commiphora mucul. And CDRI Lucknow, they also developed a drug from Bacopua muneri, which was used as a memory enhancer. Now students, there are some challenges also in the path of reverse pharmacology. So let us discuss a few of them here. The first challenge is that most of the work in this field of reverse pharmacology, it is confined to the traditional practitioners, the practitioners who are practicing the traditional system of medicine, or to the academic research laboratories. The second challenge is that we have been using these compounds for ages, right? But there is lack of proper documentation. Also, when we are manufacturing these drugs, there is lack of proper good laboratory practices and good manufacturing practices, which are the essential and cornerstone of the drug discovery process. Okay. There is also lack of financial support. It's a new concept, so there is lack of financial support. Again, there is a lot of cultural prejudice towards accepting this as a proper and an authentic form of science. And the last point is that the traditional pharmaceutical companies, they are providing lesser support. Okay, they are mostly trying to stick to the traditional methods of, that is the conventional method. But without their help, it will be very difficult to do this on a mass level. That's it for today, everyone. If you like the video, then please give it a thumbs up. Don't forget to share it among your friends. Also, do subscribe to our channel and press the bell icon so that you don't miss out on any upcoming videos. Do let us know down in the comments below what other topic you will like us to discuss. And we will meet you again very soon with more topics. Until then, goodbye and take care.